Thank you very much, Dr. Obeid. Very nice talk. Now we know that lipids can stress out cells. And when lipids stress out cells, bad things happen. So let's keep that in mind. Our next speaker is going to be Dr. Semir Beyaz. You can come up. Dr. Beyaz got his PhD in immunology from Harvard. Then he worked in Dr. Stuart Orking, who is a known person in epigenetics and regulation of cell death. And he got accomplished through that process and received numerous awards and scientific accomplishments, including the Jeffrey Models Prize. He also got prizes from the American Association of Immunologists, Training Achievement Awards, and a lot of other awards that we don't have to mention. Today, he's going to be discussing the dietary regulation of cancer and immunity. So he's going to look at the relationship between diet and how that influences the immune system that leads to the development of cancer. So thank you for coming.
All right. Thank you. Thanks to the organizers for putting this amazing symposium. There are great talks, and it's a great honor to speak after Lena. I'm a big fan of her. So today I'm going to talk to you about two stories that are seemingly distinct, but they are interconnected. And my lab at Cold Spring Harbor is trying to understand a seemingly simple but really complex question: are we really what we eat? And so this is a question that everyone can comment on, but the problem is there is very little we know about the causality of the associations that we make depending on what type of food we eat and how much they can influence health and disease states. So this is an important problem because what we eat influences not only the way that the nutrients are utilized in our body, but they can correlate with severe pathological states. So obesity is a good model to answer that question, how what we eat may influence who we are. And this problem has been becoming more and more evident both in the US and in the world. So this data is summarizing the incidence of obesity over a decade from 1994 to 2014, and as the color gets darker in red, the incidence becomes higher. And as you can see, it's a self-explanatory slide that it's becoming darker and darker. And this situation is not just limited to the US adult population, it's also influencing childhood, adolescence, and adult obesity rates in the world, in many countries. So to emphasize why this obesity problem is relevant to this symposium is because there are racial and ethnic inequities that are associated in obesity. So the disparities that are stratified by ethnicities or racial background is summarized in that slide. And as you can see, the incidence of obesity and the rates of obesity, although they are increasing irrespective of the race and ethnicity, there are some differences in rates which are highlighted here in total. In non-Hispanic, Black and Hispanic populations, the incidences are much higher compared to non-Hispanic white and non-Hispanic Asian populations. So there are lots of potential explanations as to why this is happening, and but the problem between these disparities in obesity and the overall increase in obesity rates is a significant health concern. And the reason is obesity is not a single disease, but it is associated with multiple diseases. Examples include cardiovascular disease. Here I'm highlighting the association between obesity and cancer. So many distinct organ and tissue types have significant associations with obesity. What I study is currently colon and rectum, which shows a significant association. And so to understand causal mechanisms as to why obesity or diet-induced obesity may lead to more cancer, we need to divide that question into multiple modules. So the association between obesity and cancer comes from epidemiological studies, and there are significance of these studies, but the causality of these associations are currently lacking. We don't know in particular what cells and what molecules are altering the states that leads to more cancer. And so we don't know to what extent diet is a major player in that equation. We know certain types of diets are pro-obesogenic, such as high-fat diet and Western diet, and societies like the US who consume predominantly these type of diets exhibit higher incidences of obesity, but there are also non-diet mediated influences on that. And so the one key question in my research is to also understand how diet directly can modify the risk of cancer by either altering the cancer intrinsic programs or cancer extrinsic programs. So this is just to again highlight it's a seemingly simple question, but it's a very complicated problem, and there are lots of variables and lack of causality in associations. So we modeled this obesity association to cancer in the context of colorectal cancer several years ago, and now this is a published study where we fed black 6 mouse long-term lard-based high-fat diet, and as you can see here, the mice got fat after over a year of feeding with high-fat diet, and we analyzed the cohorts of mice, and we found that in mice we can recapitulate that epidemiological link that exists between obesity and cancer, where you can see here with red the carcinoma and dysplasia incidences, and with blue is increased. And so this it linearly can be explained like that. So you give high-fat diet as an input, and you get more tumor, but the question mark is a complicated question mark. And so to understand that question mark, I would like to tell you a story. It's called the elephant story, and it's a modern day story now where scientists are trying to identify what this is, and you guys all can see that this is an elephant. So it's the same elephant that we are trying to understand what can cause this high-fat diet-mediated or obesity-mediated increased risk of cancer. And so all these scientists are here claiming this is something else based on their point of view and perspective. According to this scientist, it's a wall. So now I will tell you our side of story and how we need to alter sometimes our perspective to discover the elephant. And so to dive into that, I will introduce you briefly what do intestinal cancers actually mean throughout that evolution of the tumor. So intestinal cancers have different stages, and the cell of origin of that cancer is mostly a stem cell that is present in the mucosal epithelium that separates the tissue from the external environment. And so this is, again, you need to make a choice to understand why when you give high-fat diet to a normal, non-mutated background mice, a high-fat diet, it would give you as an output more cancer. It could be at that stage, it could be at that stage, it could be in all of those stages, but to identify a causal mechanism, you need to be a reductionist and make a decision. So here I will just show you how we study this problem. So the data so far suggesting that most of the time tumor initiation happens due to mutations that are acquired in the intestinal stem cells. And so we took advantage of the advances in this field where, thanks to Hans Clevers and many other people in the field, they identified what are the markers or like the definition of those stem cells inside the intestinal crypt. And here I can highlight to you that the cells that are labeled at the bottom of the crypt, they have that stem cell identity. They are positioned next to their niche cells called Paneth cell. They can elaborate some niche-mediated signals for their maintenance. And so the advancement in profiling these cells and the ability of these cells to be propagated ex vivo in a dish using organoid culture conditions enable us to characterize these stem cells in terms of their activity ex vivo. And so we did that, and I'll just summarize you the key point from that study is the fact that when you take stem cells from a high-fat diet, you see elevation in their stemness. And so this is summarized here. If you take stem cells alone, they are not able to propagate organoids. They need their Paneth cell niche. But when you give the Paneth cells, they are able to form proper organoids. If you take stem cells from a high-fat diet, they have the super stem cell properties. They no longer need niche-mediated signals, which suggests that high-fat diet is modifying that qualitative aspect of the stem cells. So further analysis demonstrated that not only the qualitative aspects of the stemness is increased, but also quantitative aspects of it is increased. Numbers and proliferative potential of the stem cells were much more elevated. And the key data was here the fact that when you give ex vivo lipids to those stem cells in a culture condition, that elevation of stemness was also demonstrated. So the presence of lipids, the lipid-mediated signals in the high-fat diet were probably driving that phenomenon. So to dive into that, we did lots of gene expression studies and identified a key mediator of that process. The mediator was PPAR delta. It's a lipid-sensing transcription factor. It regulates lots of processes regarding lipid biology, including fatty acid oxidation. I highlighted it here. But what also it does as a transcription factor, it can couple with multiple other transcription factors. And so a key transcription factor we identified that is coupled to PPAR delta in that condition is beta-catenin. And beta-catenin, as I showed you initially, is associated with stem cell maintenance and activity. And one of the initial oncogenic mutations that lead to colorectal cancer targets beta-catenin program. So APC is one of the earlier mutations that happens in that process. So what we found as a model is in high-fat diet, you get more tumors because your stem cells are qualitatively and quantitatively increased, and the progenitors, together with the stem cells within the crypt, have this elevated stem cell state. So when these are coupled with an oncogenic mutation, they give rise to more tumor compared to the control state. So it's just a statistical analysis that led us to speculate that this is one of the potential contributing causal mechanisms to that process. And how did we determine causality? By demonstrating that PPAR delta activation was sufficient to mediate that process, and PPAR delta inhibition was also necessary in that phenotype. So that opened up a paradigm that potentially targeting PPAR delta in that context, it can be a modality for treatment of the obesity-associated cancers. And for the health disparity standpoint, I think that merits some further discussion. And I would love to hear your points in the panel discussion. So we published this paper, and it was nice to see after our publication that focused on tumor initiation, many other publications demonstrate the significance of PPAR delta and downstream mediators of PPAR delta in other tumor models and in other stages of tumorigenicity. So both tumor progression and metastatic potential of the tumor cells were significantly dependent on PPAR delta and its mediators, and also can be enhanced in response to lipids. So that was a good part of the elephant I think we discovered, and it's right here. Now I can say, hey, it's PPAR delta. But so during my PhD training, I was lucky to get exposed to multiple different disciplines. I was trained as an immunologist. That was a side project that I got drawn into for several reasons. But what I want to tell you is when you are trying to identify the elephant, which is a very big problem, how diet modifies cancer risk, that's a very, very big problem. And as a young scientist diving into that question is, of course, ambitious and challenging, but I'm ready to take on that. And when I'm ready to take on it, I want to tell you another type of story where we can identify another part of the elephant by module dissecting the complexities. So the next story I want to tell you highlights the immune aspects in the mucosal tissues. So gut is an immune organ, and there are lots of immune cells, much more than many other parts in your body, and they're constantly interacting with the external environment that are mostly mediated by the gut microbes and the metabolites from the microbes and the food antigens. And so intestine in that complexity it cannot be studied just by looking at intestinal stem cells or tumor-initiating cells or tumor cells. So it's a complex network. So the question that I wanted to follow up was how immune surveillance mechanisms are perturbed in response to diet. So we know a lot about immune surveillance mechanisms by now, thanks to the advancements in the field of immunology. We know that in the case of tumor immune surveillance, there is one key adaptive immune mechanism in which tumor cells can present antigens either on MHC class II to CD4 T cells or MHC class I to CD8 T cells. And the current dogma is the fact that CD4 T cells, through recognition of some peptide antigens on MHC class II, can help CD8 T cells to release cytotoxic molecules that can kill the tumor cells. But many papers in the field suggested that there are other ways that you can also mediate that tumor rejection, that anti-tumor immunity, and those may involve a direct contribution to the killing by releasing cytotoxic molecules, recruitment of other cells, and through some unknown cooperation between CD8 T cells or like cytokines that may be released to also dampen tumorigenicity. So this is also on its own is an elephant and a complicated problem, but we know that at least from tumor immune recognition standpoint, tumors try to escape from anti-tumor immunity. And to do that, one of the first things that they do is they downregulate their immune recognition molecules. It's like they get rid of their identity cards and passports. So MHC class I is an identity card, and if you don't have it, NK cells can come and kill you. But MHC2 is a passport. You can stay undercover unless you're not traveling around the airports and highly surveyed areas. So we were lucky enough to see that in high-fat diet, a separate mechanism that may contribute to anti-tumor immunity was the fact that a high-fat diet led to dampening of immunomodulatory gene expression in stem cells. So those stem cells, I want to remind you, had an enhanced stemness, enhanced qualitative and quantitative and qualitative aspects in response to high-fat diet. But they also downregulate the genes involved in MHC class II pathway significantly. So we looked into that phenomenon, and since the discovery of MHC class II antigens, both in mice and in human, people have stained the intestinal tissue and have demonstrated that intestinal epithelium expresses MHC class II. So if you look at the textbook, they say MHC2 is only expressed in antigen-presenting cells, but the reality is MHC class II can be expressed in multiple cell types because it's a very responsive molecule to inflammatory cytokines. And so in the gut, we found that in control state, in control diet, most stem cells were positive for MHC class II, and in high-fat diet, this is significantly downregulated. And so the implication is downregulation of MHC class II can be losing that passport, and if those stem cells lose that passport, they can go throughout that tumorigenic evolution faster. So to test this, initially we used the organoid system to understand whether or not cell-intrinsic oncogenic programs are whether or not are altered, and we found that they are not. So we used pre-malignant cells that lost APC and sorted them out based on their MHC expression pattern and found that they are both equally clonogenic in vitro. But in vivo, we developed this orthotopic transplantation system where we can transplant stem cells to the distal part of the colon, and when you transplant MHC positive and MHC negative pre-malignant cells, you can assess their in vivo tumorigenic potential. And we did that, and the result was really surprising and exciting. We found that in the case of absence of MHC2 in a pre-malignant cell, you get much more tumor compared to the control if it is in an immune competent host. But if you do it in an immunodeficient host, that rate was equal. So this is suggesting that the presence or absence of MHC2, which is the passport molecule, is influencing the growth of the tumor depending on the availability of the surveillance system, which is the immune system. So that one is a convincing data, but to show this effect definitively, we developed a transgenic model where we deleted MHC together with loss of heterozygosity model of APC. And in this genetic model, we were able to see that there are much more tumors in the intestine in the absence of MHC compared to the MHC intact counterparts. So there is a relevance in a clinical setting for this phenomenon, and we analyzed TCGA and ICGC data for colon and rectal adenocarcinoma patients, and we found that MHC high expressors survive much better compared to MHC low expressors. So there are lots of contributing factors probably to that survival data, but at least there is a significance that would make us think there may be an impact based on tumor MHC expression throughout the tumorigenic profile. And recently we started diving into this problem deeper, and we have been developing ways where we can, without biasing the system too much, assess how MHC expression can correlate with distinct stages of tumorigenicity. And this is just a brief data suggesting that across tumorigenic program, MHC expression module is dampened. So in tumor or metastatic organoids, we see that there is less and less MHC compared to the tumor. So, but the key question that's remaining is what is the mechanism of MHC expression or downregulation in the context of diet or obesity? And I will show you just two key pieces of evidence, which is based on what we already know about how MHC is regulated. So MHC can be regulated either by pattern recognition receptor activation in response to microbial signals, or cytokine induced JAK-STAT activation. And we found that, and many other people suggested that in response to high-fat diet-induced obesity, you perturb the microbiome, and this is just showing how microbial diversity is altered in the intestine. And the antibiotics were used as a control, but the intriguing phenomenon we found here, where we stained for MHC2 mRNA using in situ hybridization, is the fact that in high-fat diet and in antibiotic-treated mice, here I can show you in the crypts where the stem cells reside, there is a dampening of MHC2 mRNAs. And so this is the control setting where you see a lot of red, which indicates the MHC expression, and here there is not much. So this suggests that depletion or getting rid of certain microbes is probably contributing to that high-fat diet-mediated MHC downregulation. And so to go deeper into that mechanism, we did microbiome sequencing to deeply identify what are the potential causal microbes in that process. And so on the left side of this plot, you can see the microbes that are depleted, and right side, it shows the microbes that are enriched. And I would like to highlight one of them, the Helicobacter species, are significantly ablated in response to high-fat diet. And the reason that I'm telling this to you is because finding a causal microbe in a microbiome study is really difficult. But I was lucky to come to Cold Spring Harbor because the mice that I used back when I was in Boston were Helicobacter positive. And at Cold Spring Harbor, we have a Helicobacter negative facility. And I have a cohort of LGR5 mice that were in Helicobacter negative, and I compared their MHC expression, because when I was analyzing it, we saw really reduced MHC expression per stem cell. Oh yeah. So in that equation, we are thinking that Helicobacter-mediated modulation of the microbial ecosystem may be contributing to the steady-state expression of MHC2, and high-fat diet is probably altering that ecosystem. I would like to highlight the complication is because we see that if you put a Helicobacter-negative mice on high-fat diet, there is also some downregulation of MHC2. So I think it's good to consider microbiome as an ecosystem rather than a single entity, but at least we have a clue into that ecosystem that Helicobacter may be playing a key role. So to definitively assess these processes, we have been utilizing germ-free mice and doing germ-free add-back experiments and also doing some mechanistic experiments focusing on the PRR signaling and cytokine signaling. But I want to highlight to you basically in germ-free mice, we see this dampening of MHC2 expression. And when you colonize some candidate microbes that we may think modulating MHC expression, we see the elevation. So to summarize, we are proposing that this tumor initiating stem cell immune recognition through MHC expression plays a key role in anti-tumor immunity, and in high-fat diet, due to dampening of MHC2 in tumor initiating cells, the immune system cannot recognize this properly, and the dampened immune recognition, it's known that it can break the anti-tumor immunity in multiple aspects. We generate those tumors that we are seeing, and I think this is an orthogonal way that high-fat diet can lead to more tumor, and the data is suggesting that the maintenance of MHC2, that passport molecule, is dependent on a healthy microbiome. We don't know what it is yet, but it looks like Helicobacter species are doing something in that equation. And an altered microbiome induced by the diet is doing something that we think it involves a cytokine pathway and also a PRR pathway. And now we are digging deeper into that understanding. But in summary, this suggests that diet-induced alterations in intestinal microbiota can influence the immune recognition abilities of tumor-initiating cells, and that mechanism can be also a potential key causal mechanism behind this obesity-associated cancers. So now we have another part in that elephant story. We thought it was PPAR delta that does it all, but it looks like MHC also can do it all. So the reason that I told you the elephant story, because we need to be reductionist when we are doing the experiments. You need to choose something and go deeper in that aspect, but also it's good, especially with problems like diet association with cancer or diet association with immunity. These are really convoluted problems, so we need to take a module by module approach while keeping an eye on the other contributing factors. So in my lab, we are trying our best to do that. It's not an easy thing, I can acknowledge to you. So the approach we are taking, dividing these different modules and assessing the phenotypes one by one. So I showed you an example how we can use nutrients such as fatty acids to perturb intestinal stem cells and understand how nutrients can influence tumor initiation processes. We have a huge interest in understanding how nutrients are influencing immune intrinsic mechanisms. I'm giving you an example how nutrients can alter T cell states, and T cell states by mean T cell migration, T cell activation, T cell memory. And so the black box, which I don't know if I will have enough time to uncover how exactly these things are happening, because we don't have the tools to definitively assess how diet or anything else that can alter the microbiome can causally influence phenotypic states. It's a very hot, popular area, but I think we need to also critically evaluate that area so that we can force innovation to create new tools, new models. So I put the microbiome in the end because I really don't know where to put it because it can influence everything and can be influenced by everything. So with that, we are trying to use this input-output module, so use nutrients as inputs, and then use perturbation through assessing how these things are altering each module, and then phenotypes that we study, I call them the outputs, and some of them can be relevant therapeutically, as I showed you in the context of targeting PPAR delta or elevating MHC2 expression, and the end goal is to build a unifying model to uncover the elephant. And with that, I would like to thank people in my steadily growing lab. So Mike left, and he was working with me for 3 years now. He's now a graduate student at Harvard, and Miriam, Highway, Onur, and Brian are my current members, and each of them are diving into a separate question, and hopefully synergistically we will uncover the elephant. I would like to thank Doug. He's my mentor and the leader of the growing immunology community at Cold Spring Harbor. We do some work with Tucson Lab on the organoid models, and Tobias is a recent faculty recruited from UK, and we have been also thinking to expand our research objectives, and I have several other former and current collaborators I would like to acknowledge, and I would love to take any questions, I think, after the, yeah, okay. Well, thank you so much for your attention.
Thank you very much. So we would go to our next speaker, but just wanted to say that that was a great talk too as well. So now you can begin to understand how diet actually influences cancer. And this level of detail is really very important for us to understand the underpinnings of these processes so that we can identify the right interventions and studies to mitigate the cancer issues.